We've reached a very important section of our Magee Summit, which is the prize competition for the three best projects. So I just want to say a few words about it before we start with our presentations. We have done the Magee prize competition now. This is the third time, so we've done it at each of our Magee Summits. The idea has been to support an award for 2 or 3 years of collaborative research between an investigator here in Pittsburgh at Magee-Womens Research Institute and somebody else either in a country or across the world with an idea of forming a new collaboration that did not exist before on a project that is transdisciplinary, maybe out of the box, tackling an important biological question. Could be basic, could be translational, could be very clinical or health services. And we wanted it to be novel. We wanted something that is going to be hard to fund by the more traditional funding mechanisms. And we wanted something that's going to be introducing innovative concepts or innovative methods and some other new drugs or potentially new approach, primarily emphasizing high risk and high reward strategies. This award has been funded by the Richard King Mellon Foundation, who have been also funding our summits to a large degree, and we're really grateful for the RK Mellon Foundation for doing this. So in addition to forming the networks, we're trying to really instill new research ideas that can stimulate new trajectories here in Pittsburgh and also hopefully across the country and across the world in the field of either early human development, women's health, or broadly reproductive sciences. This year, we've received 16 applications. There are more questions, letters of intent, but ultimately we received 16 applications. 13 led to more complete application to be reviewed by our referees, reviewers who are really across the world. We had 2 cycles of reviews. One cycle was what we call a triage review. Reviewers just gave us a serious but not point-by-point reviews and gave us a score. This was just the idea to get a triage assessment and to lower it down from 13 applications went out to 6. And then the 6 best applications received reviews by each application by at least 3 reviewers, very thoroughly from reviewers across the world. And then we put it together to the 3 best applications, and they're going to be presented now here. So first, I'm not going to mention the names of the reviewers, but I do want to thank all of them. Some are here, some are elsewhere. But I do want to thank all of the reviewers for the thorough work, and I also really want to thank Lori Rideout and Katie Chapa, who's going to be putting the scores together, as I'll outline in just a minute. We have 3 prizes for the Magee Award. The third prize is going to be a $4,000 prize that's going to be shared by the co-PIs. It's a personal prize for them to be used in any way they want. For the second prize, we give $6,000, again, to be shared among the co-PIs. And the best prize is $1 million, including $18,000 to be shared among the PIs and co-PIs, and the rest of the money goes for the research grant, as we heard this morning from the best presentation by Pam Moali, Carolyn Gerget, and Kyle Orwig. So how does it work? In just a minute, we're going to have the 3 presentations. Each one of them is going to be for 15 minutes. We will not have any questions and answers, so it's going to be exactly 15 minutes. Some of the presenters are here. Some will come to us with video. We'll see how it goes. And ultimately, at the end of the 3 presentations, we're going to have a QR code here, and you'll be able to scan it. And only those people who register for the meeting, and you need to use your name and email the way you registered for the meeting for that. And I'll explain just in the end of the presentations. And that's going to lead us to a vote. It's going to be an electronic vote. We'll vote for each of the 3 applications, and I'll explain it when we vote. And then that score is going to be factored in with the existing score from the second level of very thorough review, and the results of that score But what we do tonight or this afternoon, and also the scores that we have up till now, will be combined together. And then by the dinner time tonight, we're going to have the ultimate scores, and we'll be able to announce the third place, the second place, and the first place. So unless there's any burning questions about it, and I'll explain when we get to the vote part, I'll explain. We'll go ahead and start with the presentations. The first presenter is Ron Bukhanovich. And Ron is a professor of medical oncology in the Department of Medicine and also OB/GYN reproductive sciences. He's the co-director of the UPMC Hillman Cancer Biology Program. He's also a co-director of the Hillman Women's Cancer Research Center here at the University of Pittsburgh. The choice of Ron is just coincidental. We went alphabetically, so Ron is a B. Please go ahead, Ron.
All right. I am super excited to be here to present our Magee Prize proposal, understanding a link between obesity, high-risk endometrial cancer, and potentially racial disparities. This is on behalf of my co-PIs, Dr. Victoria Bay-Jump and Dr. Bo Roida. All right, so we often start, as oncologists, we often start with doom and gloom. I'm going to start with good news. And so the good news here is if you look at this slide, cancer death rates have actually dropped over the last 20 years. And this directly correlates with when we decided to invest in funding, the war on cancer. We invest in funding and we improve outcomes, right? So this is an important take-home. When we invest in cancer research, we can improve lives and improve outcomes. Okay, I am an oncologist, so next to the doom and gloom. The bad news, uterine cancer. So these are the rates of uterine cancer over the past 20 years. And I can't really see the screen here, but you can see that uterine cancer rates have been going up, 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 right? Even worse than that is if you look at mortality rates. So not only are we seeing more people develop cancer, but the number of people dying per 100,000 is actually increasing as well. So this curve is sliding up. And why is that curve going up? The curve is going up because we're seeing more high-risk uterine cancers. So we're seeing uterine cancers that are the type that present metastatic or the type that don't respond to chemotherapy. So we're seeing high risk, the more deadly uterine cancers. And even worse, we're seeing these very disparately. So this curve represents black women. This curve represents the rates in white women or Hispanic women. And you can see the rates are much higher in black women than white women. And we don't know anything about why that is. So with the rising rates of uterine cancer in the United States, uterine cancer this year, in 2024, passed ovarian cancer as the most deadly gynecologic cancer. So I've dedicated my life to ovarian cancer. It just got passed by uterine cancer. Furthermore, because of the rising rates, it will pass colorectal cancer, and by 2030, it will be the third most common cancer of women, right? And we don't know anything about uterine cancer. Sorry, my clicker's not working there. So let me go back. Why are rates going up? So I show you the rates, the incidence on the left, the rising rates. So the rates are going up in part, we think, is if you look at this curve, keeps doing that. If you look at this curve, look at this curve, the uterine cancer rates are going up, and they mirror the rising rates of obesity in the United States. So obesity is becoming endemic, and uterine cancer rates parallel that. So if we look at obesity and we look at uterine cancer rates, one of the things we know, so a measure of obesity is something called BMI, the body mass index. So if you look at the BMI and you look at the rates of BMI in that curve on the left, so you can see that there's a mild increased rates of things like breast cancer. Doesn't really impact ovarian cancer. If so, it does really modestly. But endometrial cancer rates skyrocket. With increasing obesity, we see a significantly rising rates of endometrial cancer. Furthermore, on the right, this is showing you the relative rate of dying based on your obesity, your body mass index. And so if you have a higher body mass index, here, uterine cancer is by far the worst. So obesity and uterine cancer are highly linked with increased death rate, right? And so people worry about it in breast cancer, but that's way up here. Uterine cancer rates are much, much more deadly when linked for body mass index. So our Magee Prize questions are these. What are the drivers of these obesity-associated high-risk uterine cancers? right? And if we know the drivers, can we identify some novel therapeutic approaches specifically for these high-risk tumor types? And can we develop a biomarker? These are ambitious, but I believe science should be ambitious. All right, so for this, so the Magee Prize, when it was first announced, was supposed to be a new research direction. I told you I'm an ovarian cancer scientist. I've been for 20 years. So this is a new direction for me. Now, in order to go a new direction, I could spend the next 20 years developing all the tools I need to study endometrial cancer or uterine cancer, or I can collaborate. It's much easier to collaborate. I need to retire in 20 years. So I put together, the great thing about this with the new direction is you can reach out, you can pick the best people you can find, right? So I have two outstanding co-Is that I'm going to work with. Dr. Bo Reuter from the Mass General Hospital. Bo has an incredible set of all the tools we need to study endometrial cancer. And you'll hear from Bo in a few minutes. And then Dr. Victoria Bay-Jump, who is one of the leaders in uterine cancer disparities in the world. And so really an outstanding team. And so we're going to focus our research on this gene EGFL6. And EGFL6 is a driver. And I'm going to tell you why. So EGFL6, first of all, we know EGFL6 is linked with obesity. So EGFL6 is known to be expressed in fat. EGFL6 levels go up when people gain weight. Their body secretes EGFL6, and the levels in serum will go up. The curve on the left there shows that when patients lose weight from left to right, the EGFL6 levels go down. EGFL6 regulates stemness, and we know stemness is critical involved in cancer. And so what do we know about EGFL6 in cancer? Well, we've been studying it in ovarian cancer, and we know that it drives ovarian cancer stemness, it drives metastasis, it drives blood vessel growth. Dr. Sandra Cascio, who's here in the audience, just published a beautiful paper showing the role of EGFL6 in regulating the immune system to drive immune suppression linked with cancer. EGFL6 in uterine cancer, when we look at its expression in the tumor genome atlas, you can see that EGFL6 expression is primarily linked with high-risk uterine cancers versus the low-risk uterine cancers. So we know EGFL6 is up in this subset we'd like to target. And consistent with that, when you look at EGFL6 expression across uterine tumors, what you can see here in the pink curve is that when tumors make high levels of EGFL6, those patients who have those tumors with high EGFL6 expression, they do much worse. Their survival is much worse. Whereas the patients who have low EGFL6, you can cure those patients. It's those patients in pink, the EGFL6 expression ones, we can't cure right now. We don't have good therapeutics for them. So we're interested in EGFL6, and I'm glad it's not dinner time since I'm showing you this. But EGFL6, we were interested, so we turned it on genetically in a mouse. And so when we did that, what we saw, surprisingly, not what we expected, if you look at the curve on the left, this is the weights of the mice over time. And so what we saw when the mice, when we aged them out, the black dots are all the EGFL6 mice, and the red dots are the control mice. And what you can see is those black dot mice, they're much heavier. They gain a lot of weight, like a lot of weight. And so this shows you just some of the pictures here. You can see this is a healthy mouse. This is an EGFL6 mouse. You can see it's about 50% bigger. And when we open that mouse up, when we euthanize the animal, it's morbidly obese. You can see all this is fat tissue. A normal mouse doesn't have barely any fat, right? Mice eat all good high-fiber diets, right? But these mice get morbidly obese, and they're on a normal diet. We're not feeding them the Western diet that we usually have to do for diabetic mice. These mice are getting massively obese on their own. Now, if you look at that curve, what you can see is some of these mice gain weight, and some at around 12 to 18 months start to lose weight again, right? And when we euthanize some of those animals, what we saw was, first of all, when we euthanize them at about a year, is that the uterus looks abnormal. So the uterus starts to have all these blood vessels feeding it, right? That's angiogenesis, growth of new blood vessels. And that's what we see commonly in cancer. In order for cancer to grow, it needs blood vessels, and it needs it to feed it. And so this is abnormal. This is a mouse that's postmenopausal. It should not have all this vasculature. But if we wait till around 18 months, what you see is this. This is a big nodular tumorous uterus, right? And so when we take that out, this is immunofluorescence for cytokeratin 7, which is a marker of the epithelial cells. Normally, the cytokeratin 7 positive cells in a normal uterus should be like these small round circles in the upper left of this red slide. But what we see are these big, disjointed, crazy growing cells. This is cancer. When we look at those cancers, they express the protein P53. So P53 is when it's nuclear and abnormal like this is a marker of the high-grade deadly uterine cancers. So we are generating high-grade deadly uterine cancers. And you can see the P53 here in brown. When we look at these mice, for the first ones we sacrificed, we've got about 30% with uterine carcinomas. We have carcinosarcomas and adenosarcomas. We see a few other obesity-associated cancers as well, including hepatocellular carcinoma. But we're seeing this huge uterine phenotype. So the first aim of our Magee Prize is to understand the tissue of origin for EGFL6. So the mouse that I just told you about is a little bit aberrant. It's a little bit atypical in that we turned EGFL6 in all cells in the body. And that's not normal. Normally, EGFL6 is made in the fat, and it's made in the connective tissue, and a little bit in the blood vessels. And so what we want to do is express it in the right tissues. Importantly, we've actually already started this. Yaki Barak had an adiponectin Q-Cre mouse here at MWRI, and he was generous enough to donate those mice. We turned EGFL6 expression now on in the fat, just in the fat. And when we do that, what we see is, lo and behold, these mice gain weight. And there again, so at around 18 months, these mice are almost double the weight, right? A 70-gram mouse, that's huge. A normal mouse is 35 to 40 grams. So these mice are very heavy. Ooh, let me go back. Importantly, sorry, it's a little bit jumpy here. We've euthanized, so just in order to present today, we euthanized a subset of just 6 mice to look and see, and 4 of the 6 mice had a uterine phenotype, either complex atypical hyperplasia, the uterine cancer precursor, or full-blown frank metastatic cancer with a metastasis in the abdomen. So these mice are having a uterine cancer phenotype. Specific aim 2 of our grant is, so now that we know we're getting these uterine cancers, is to see can we understand a therapeutic phenotype. So what we did is we studied them, we've looked at some of these early tumors, and we characterized them. We also knocked this EGFL6 out in cancer cell lines. And what we see is when we knock EGFL6 out, we lose expression of a gene HER4. So HER4 is a cousin of HER2, which is the receptor for Herceptin. So think about breast cancer and what Herceptin, or the Herceptin target has done in breast cancer, right? It's revolutionized care for women with HER2-positive breast cancer. This is HER4, cousin of HER2. So we think this is an important therapeutic target. And what this says is that EGFL6 is likely regulating expression, and it's via this gene LXS1. I won't go into the details there. When we look in the tumors, and this was work of Dr. Shumei Bai, when we look in the tumors, what we saw is this gene, NRG1, is specifically upregulated in EGFL6 expressing tumors. What's interesting about NRG1 is the ligand for HER4, right? So now we're turning on both the ligand, right, and the receptor. So we're activating this system. And when we look at this, if we take tumor cells and we treat them with EGFL6 alone, NRG1 alone, we don't see a whole lot of effect. But if we add both of them, they work together and they work synergistically, and we're seeing this huge increase in tumor cell proliferation when we add the two together. And so in order to study this, this is where I need the systems. I need to have these models, and this is where my colleague Bo Reuter comes in. And I'm hoping, I don't know how to turn that video on.
Good afternoon. I'm very much looking forward to the opportunity to work with Drs. Bukanovic and Magee on this exciting and impactful project. As you just heard, obesity-associated uterine cancer is an area of urgent and unmet need. When Dr. Bukanovic reached out and shared his vision of forming a new team of investigators with different expertise and resources to tackle this significant clinical challenge, I jumped at the chance. His findings indicate that EGFL6 may be a critical link between obesity and uterine cancer suppression, just beg for us to define its role. So how do I fit in? To do a deep dive, you need relevant models. My team brings in some expertise in endometrial cancer and unique resources. Specifically, we have an established tissue repository for which we develop clinically applicable models to better understand the underlying biology related to the genesis, progression, metastatic potential, treatment resistance, and recurrence of gynecologic cancers. Following informed consent, we collect benign and/or malignant samples and correlate biologics. These include blood, ascites, pleural effusion. We take these back to the lab for processing for model development, distribution, or cryopreservation. The models include organoids which resemble tumor spheres. We can also generate xenografts in mice. These are also called PDXs, for which we can expand to have mini clinical trials. All the models are open for genetic and functional interrogation. We can also genetically manipulate these models to define their contributions in different environments. In addition to these, we have also adapted syngenetic models of endometrial cancer. In summary, my team and I are committed to this collaborative research effort and confident that together with my colleagues and your support, we will make a difference in the lives of our mothers, sisters, daughters, and their families.
Excellent. Thank you, Bo. All right. So I'm going to tell you now about the third aim of our proposal, and that is to understand the role of EGFL6, potentially linking it to disparities. So when we saw that EGFL6 was linked with these high-risk uterine cancers, the type we're seeing are much more common in black women versus white women, we were able to reach out to colleagues at MWI and UPMC, and we collected some fat tissue to say, what if EGFL6 expression is higher in black women than white women? And so we looked, and what you can see, I hope you can see here, this is, first, this is a curve which just shows that with increasing body mass index, we see increasing levels of EGFL6. This is good because this replicates what other people have shown, and it means our assay is working. But what you can see if you look in the bottom, when we do this, so the white dots are white women, the black dots are black women. What you can see when we match patients for pound, what we can see is there are some black women and white women who all have EGFL6 levels that are lower, about below 10. But then there's a subset of black women who have very high EGFL6 levels in the 30 to 40 realm. And that's regardless of body weight. And so, for example, here is fat from a woman, a black woman who has a BMI of 44, and the EGFL6 is stained in brown. This is the fat from a white woman with a BMI of 50, so she's heavier, yet her EGFL6 levels are much lower. So what we would hypothesize is EGFL6 could be a biomarker of this really bad disease. So imagine, I told you EGFL6 makes more fat. And so if there is somebody who's epigenetically or genetically prone to make more EGFL6 in their fat, they make more EGFL6, that makes more fat. More fat makes more EGFL6, and that's going to be a feed-forward cycle. So now you've got a very high EGFL6 level, and that's going to make you prone to these high-risk uterine cancers. So to ask this last question, we go to my colleague, Dr. Victoria Bay-Jump.
I'm honored to be part of this study team and this exciting project. What I find so exciting about this project is that EGFL6 links for the first time obesity, which is a known driver of endometrial cancer, with our most aggressive molecular subtypes of endometrial cancer and disparities for Black women battling this disease. This project will take advantage of the Carolina Endometrial Cancer Study, which is our UNC population-based study that has the goal to address racial disparities in endometrial cancer. The leadership of our study team is shown here, although there are many amazing folks involved in this project. In brief, the Carolina Endometrial Cancer Study is a North Carolina-based population study that will reach all 100 counties in North Carolina. The goal is to oversample Black women as they have been largely underrepresented in genomic studies and clinical trials, with a plan to enroll 1,800 women overall and greater than 500 Black women. The study opened officially in February 2021 with approximately 700 plus women enrolled to date, 230 plus Black women. This study entails the collection of participant surveys, medical record data, tumor samples, and ongoing follow-up with the mission to integrate epidemiological factors and tumor biology as contributors to worse outcomes in Black endometrial cancer patients. Ultimately, we hope this work will build a comprehensive picture of this disparity and delineate the best interventions to address. In essence, the Carolina Endometrial Cancer Study is the ideal study in collaboration to build on the exciting and promising preliminary data of EGFL6 as a critical factor related to racial disparities in endometrial cancer. Thank you.
All right, thank you, Vicki. So just one and a half slides. So I told you earlier that when we invest in cancer research, we can improve outcomes and save lives. This is the investment of the NIH in uterine cancer. So you can see here, this is breast cancer over the past 6 years, $4 billion in research. Lung cancer, almost $3 billion. Colorectal cancer, almost 1.6 billion. This is uterine cancer, 112 million. 1/15 to 1/40 of these other tumor types. I told you this is going to be the third most common cancer of women. And so this is a chance for the Richard King Mellon Foundation and MWI to invest in uterine cancer, to push the envelope. I think we have an outstanding team. We have really innovative, important questions, and I think this can be high impact reward. And I'll say thank you, and I'm running over, so I'll stop there.
Thank you, Ron. If anybody has any questions to Ron, they can ask him later, but we will continue with our next presentation, as I said, alphabetically organized. Our next presenter is Carlos Escudero Orozco. Carlos is coming to us all the way from University of Bio in Concepcion, Chile. Welcome to Pittsburgh. Carlos is a professor of basic sciences at the Faculty of Basic Sciences at Bio, and he's also has his own vascular biology laboratory, and he's going to focus on the role of placental small extracellular vesicles in disrupting the blood-brain barrier and impairing cognition in women with preeclampsia. Carlos?
Okay. So I don't want to have troubles with the slides, hopefully. So thank you very much for the introduction, and also good afternoon, everyone. I'm Carlos Escudero. I came from this long and narrow country in South America, where I've been working in pregnancy complication for many years now. And during this journey, I have the opportunity to meet James Roberts, and I have the chance also to visit the Magee several times. And in the Magee, I have the chance to interact with the researchers and also work with them and collaborate with them. So this grant actually is the beginning of this collaboration, started with this collaboration with Magee. And I'm going to talk about the brain and preeclampsia. So the first thing that I'm going to tell you is that preeclampsia is a condition characterized by high blood pressure during pregnancy and also several organ disturbances. So, but one of the important things is that preeclampsia is the killer of mothers and babies, especially in low and middle income countries. But also, not only that, it's also relevant to know that preeclampsia is also the leading causes of maternal mortality in the USA when we analyze the high-income countries. But this is a big but here, because despite as terrible as it is, maternal mortality and newborn mortality is just the tip of the iceberg. Because underneath this tip, there is many women who actually survive preeclampsia. And those women have higher risk to develop chronic diseases, including heart attack, hypertension, dementia, or cognitive impairments. So nowadays, there is more and more evidence that indicate that women who survive preeclampsia actually have higher risk to develop dementia, 2 to 3 times to develop dementia, and also cognitive decline, and also impaired memory and learning processing. So one of the important messages here that I'm going to tell you is that preeclampsia doesn't end with pregnancy. Instead, preeclampsia has a long-lasting consequences for a woman. Another important message is that women who survive preeclampsia actually have higher risk to have this life-threatening condition that is cognitive decline. And one important message is that people in general, and even some clinicians, especially in lower middle income countries, and the patients doesn't know about this risk. So we have to do something in order to overcome these limitations. So this is why we decided to put this grant together to investigate the root causes of acute learning and memory dysfunctions in women with preeclampsia to identify new therapeutic interventions. So let me just dissect a little bit more about these ideas. So when we talk about the brain in preeclampsia, we are talking about different entities that affect this organ. So this affection is going to occur immediately. I'm sorry. Go back. So this affection is going to occur immediately during pregnancy or immediately after delivery. And we call them short-term complications. And you can see here there is many entities here, included, for instance, stroke, which is the most lethal condition that actually is responsible for the maternal death. But also these other entities, give some sequela for the woman who survived preeclampsia. And in this part of the slide, you can see also other entities that affect the brain after delivery, years after delivery. And the thing is that we don't know many things about these affections. We don't know how these affections occur. And we don't know what would be the relationship between this affection with the short-term complication with the long-term complications. So we need to do more about that in order to understand that conditions and also in order to prevent it. But one of the commonalities that all of these conditions have is the affections of the blood vessels in the brain. So let me just zoom in the blood vessels of the brain. So here you can see, this is just a cartoon to indicate how complex are the blood vessels in the brain, because they are highly specialized, and one of the specializations that they form these multi-layer cells that form one of the most strong barriers in the human body, that is called the blood-brain barrier. And this barrier, you can see here, is the protector of the brain. So any potential damaging elements that are capable to enter to the brain are blocked because of this barrier. So one of the important messages also is that when this barrier is broken, so this alteration of the barrier precedes to the cognitive impairment observed in people who suffer Alzheimer's disease. So many people, included some of our studies, has been analyzing the integrity of the blood-brain barrier in women who suffer preeclampsia. And we know now that this barrier is broken in women who had preeclampsia. But still, we don't completely understand why is happening that. So one of the key discoveries that we did as a group is that we found that the placenta, these interface organs between the mother and the baby, is releasing some factors to the maternal circulation, and these factors are capable to reach the brain and impair the blood-brain barrier. So if we want to prevent the cognitive decline of this woman, we must understand how to prevent this damage of the blood-brain barrier. So let me explain a little bit more about the mechanism why this barrier was impaired according with our studies. So here, I'm sorry. So it's a video here. I don't know, can you? Oops. No, it's not running the video. I don't know if you can put it in. So anyway, yeah, okay, thank you. So this is the baby. The baby is connected with the placenta, and the placenta is in direct contact with the maternal bloodstream through this space, intervillous space. As you can see, the placenta is releasing several factors directly to the maternal circulation. And among these factors, we are interested in one tiny cell fragment called extracellular vesicles. And we are interested in one kind of extracellular vesicles called small extracellular vesicle, or small EVs. And these vesicles is important because they have inside proteins and nucleic acid contained that make them signaling particles among different and distant organs, in this case, the placenta and the brain. So these particles are capable to reach the recipient cells and release the content and change the function of that recipient cells. And the cells that receiving this message are complex, such as the immunological cells, but also complex as the cells that constitute the blood-brain barrier. So what is important also to mention is that women who had preeclampsia had higher levels of these extracellular vesicles. And we know as a group that these vesicles are capable to reach and impair this barrier in the brain. So we have developed the techniques to measure them, to isolate these vesicles, to quantify them, and also to investigate the content of these vesicles, using sophisticated techniques. So let me show you just one example of these results. So here you can see a slice of the mouse brain, and in red is the vessel, the one blood vessel. And in green, I don't know if you can see the green staining here, but there is some green inside the vessels and some green outside the vessels that has leaky from this, brain barrier. So that doesn't normally happen. So what is relevant to mention at that is that despite we haven't collected all of this information regarding the the mechanism of releasing the extracellular vesicles and reach the blood-brain barrier, we still don't know what would be the meaning for the mother to have placental material inside the brain. So we need to understand what would be the consequences of that. So this is why we decided to put some complex and challenging questions in this grant. So one of the questions is, what would be the main target cells to receive this message from the placenta? And more importantly, what would be the consequences for that cell, to change the function? And also, it's important, to understand whether the placenta extracellular vesicles are capable to induce learning and memory problems in a mice model. So then we want to combine these basic science methodologies in order to also add some knowledge using samples from women who had preeclampsia and cognitive impairments. And we want to track the content of these extracellular vesicles from the placenta into the circulation and also inside the brain. So at the end of the day, we want to have the knowledge to develop therapies to save mother's life and also protecting mother's brain. Of course, these questions are complex and need one of the most outstanding group of researchers that I've been working with. So let me introduce this group. So we gather people from different parts of the world, and we are a global group, we are a global group for a global problem. So this group included Janet Katov, James Roberts from Magee, also Lina Bergman from Gothenburg University, Manu Batich from Oxford University, Jeffrey Penny from Manchester University, and myself from the University of BioBio. And also include both young researchers, both from the US and Chile. And altogether, we have the expertise to study these complex questions. We have the expertise to study preeclampsia, the placenta, extracellular vesicles, cognitive impairments, and also the blood-brain barrier. So, but more importantly, we also are including samples from women, precious samples from women who actually have preeclampsia and cognitive impairments. And these samples are coming from Sweden, from South Africa, from the UK, and from the USA. So making this effort truly global. So at the end of this study, we want to make foundational discoveries in order to understand what would be the consequences for the mother to have this placental material inside the brain. And also we want to understand how to prevent it, because otherwise all of this knowledge is going to be useless. So as you can imagine, this grant means many things for the people involved. For the woman who actually we want to make some effort to help them. For the team that has been working hardly for many years now, and also for myself. Having the opportunity to lead this outstanding group of researchers worldwide in a topic that is in the frontier of the knowledge, with the possibility to actually do something for making progress for women's health is truly meaningful. But also at the personal level, because this grant means a reconnection with my mother. So my mother was a wise woman, and she must be, because she has 9 children, she has to deal with 9 different personalities, 9 different troublemakers, and actually she said, I know every one of my children. And I like to think that with this phrase, she gave me some clues to understand how much we changed our mother's brain. So thank you very much for your attention. I engage you to vote for this grant because that's going to give you the opportunity to continue working in this field and make some progress for saving maternal brains. Thank you very much.
All right. Again, thank you very much, Carlos. And any question to Carlos, you can ask him during the breaks and the subsequent sessions. Our third and last presentation is by Julie Rios. Julie is an associate professor for OBGYN and reproductive sciences. And she's also the director of our Division of Reproductive Endocrinology and Infertility here at the University of Pittsburgh Magee-Womens Research Institute. The title of Julie's presentation is Next Generation In Vitro Fertilization, Unveiling Optimal In Vitro Maturation Protocols for Immature Oocytes. Julie?
Hi, good afternoon, and thank you again for your engagement all the way through the day. It's really exciting to see so many people show up in such a big way for women's health and women's health research. So in any case, I'm Rebecca Flicht from University Hospitals of Cleveland. I'm the division director of fertility there, similar to what Dr. Rios does here. And it's our pleasure to share with you the research that we've been collaborating on, both with my institution about 2 hours away in beautiful Cleveland and here at UPMC, as well as with our collaborators in Scandinavia. And so we're going to share with you some details about our proposal for next generation IVF and what we can do today with direct clinical applications to help improve fertility care for women around the world. Looking around the room, and it is a little hard to see, I imagine that, that many of you are parents or hope to be parents someday. I can definitely share with you my experiences in becoming a parent with all of the joy and hope and excitement, and for anyone that's ever lived with a teenager, sometimes the struggle of parenting children in this world is one of the most rewarding, one of the most meaningful experiences of my entire life. And so if you think about for patients with infertility, and unfortunately that's 1 in 6 of our patients, what they go through is a profound emotional experience. And when you look at research, looking at women with infertility, they rank that experience as being as stressful, as painful as being fired from your job, as going through a divorce, having to move to another country, even being diagnosed with cancer, which I know that we've talked a lot about today. So that tells you a bit about the psychological impact of infertility and how heavily it weighs on our patients. But that doesn't even account for the financial impact. Any fertility patient can tell you that IVF is expensive, but if we look globally, there's 2.5 million IVF cycles performed a year around the world with a total cost that exceeds $15 million. So there's a psychological impact and there's clearly a financial impact, and I think any of us working in this field would say that this is a global health issue. So looking specifically about the patients that are particularly impacted. So this is probably more than 1 in 6. We've decided to focus our research really on 3 different populations that are disproportionately affected. So let's start, again, we talked a lot about cancer today, with our cancer survivors. We are so lucky that we are living in a time where the 1.3 million children that are diagnosed with cancer can actually expect to be cured of their disease for the most part and go on to live long and healthy lives. So a key concept in oncology right now is survivorship, and we're both very involved in survivorship programs. But the problem when we get to the survivorship stage is that the same life-saving treatments, the chemotherapy and the radiation that we use to cure the original cancer, can leave a lot of these children when they reach adulthood completely infertile. And we're really lucky. We have fertility preservation programs. We can freeze eggs, we can freeze sperm, and all of that is on the cutting edge of fertility preservation. But unfortunately, a lot of times these kids are too sick to do it, and there's not time, or unfortunately, these are also very costly. Cancer survivors are not the only patients who experience increased rates of infertility. So the other 2 populations that we want to look at are patients with endometriosis. So these endometriosis patients are patients we see every day in our fertility practices. And for those of you that are not familiar with the day-to-day of endometriosis, it is a debilitating and painful disease that often results in multiple pelvic surgeries. And endometriosis surgery has been a huge focus of my career. I do hundreds of surgeries a year. And unfortunately, when we do surgery to remove the disease from the ovaries, we often significantly decrease what's in the tank in terms of someone's egg supply, which then just, promotes this cycle of infertility. And then the last of our goals really is a population of women who we consider older women, which in our world is probably anybody who's over 35. Sorry. But in any case, one of the biggest puzzles that we have yet to unlock in reproductive medicine is the fact that as we get older, our ovaries get older, and our eggs get older. And when we look at women with infertility, the rates of pregnancy drop precipitously once we're older than 35. So sorry for the audience. These were always the slides that made us cringe when we were in medical school and residency. But the sad fact is that we start losing eggs from the minute that we're born. And once we get to our 30s, our fertility starts to face a steep inflection point, which really gets significant at late 30s, early 40s. And you can see alongside that drop in fertility, it comes an equally significant uptick in the rates of miscarriages, which are mostly due to chromosome problems. I see these patients in my office every day, women who are in their 40s trying to get pregnant. And the fact is that a lot of times they arrive at a decision for IVF in their 40s, and they say, like, okay, we'll just do IVF. IVF is not a magic bullet. Even when we do IVF for patients in their 40s, success rates might be as low as 5 to 10%. And I think patients are really disheartened to hear that. Looking specifically at eggs, and that's really the focus of the work that we're proposing for our moonshot here, I think that the sad reality is that a lot of the eggs that we get in any of the treatments that we do are immature. And what that means is from a nuclear perspective and a cytoplasmic perspective, those eggs are not usable, they're not fertilizable. And so there's an incredibly inefficient process that goes with the IVF where we just don't get enough mature eggs. And so what you can see in the graph at the left is that as we get older, we have fewer mature, fewer usable eggs from some of the treatments that we do. And on the graph on the right, alongside that, we need more eggs to get to a live birth because just the health of those oocytes is not the same. And we see that in our embryology labs every day. So what do we do today? What's available for patients either who have cancer and their fertility is threatened, or for our fertility patients who have endometriosis or advancing age? Well, really the most powerful tool that we have is IVF. And IVF is basically the process of taking 2 weeks of painful injectable medications to try to grow more than 1 egg of the ovary. So we hope for maybe a dozen or more. Then they have to go through a surgical procedure to collect the eggs. And then we can either freeze them if they're facing cancer and we want to use them later, or we can fertilize them with sperm, make embryos, and implant those embryos into the uterus if they're in a position to become pregnant. And then we wait and hope. Now, that's not the only option of fertility preservation for patients with cancer. And so, over the last few decades, there's been considerable refinement of a procedure called ovarian tissue freezing. There's an ovarian tissue freezing program here. I've built 2 ovarian tissue freezing programs at 2 hospitals. And this is really the cutting edge of what we can do for prepubertal girls. Those girls haven't gone through the maturation of the hormone axis to be able to do injectable medications for IVF. And so instead, we'll do a surgery. You can actually remove an ovary. We can prepare it in the lab, which is what you see here. We cut it into thin slices, preserving the cortex, which is that outer layer of the ovary that contains the eggs. And then we can freeze that. And then later, and this could be 15 years, 20 years later, that same cancer survivor can have their ovary reimplanted. And that's actually how Dr. Rios and I first met, and how we met Dr. Klaus Andersen in Denmark, who's our collaborator. Ours is a very small world of ovarian tissue freezing. And the 3 of us have all done research so far independently of how to optimize protocols around ovarian tissue freezing to give these girls with cancer a better chance of becoming mothers someday. So I know all of that probably sounds really exciting and really great, but the fact is that what we have today is not satisfactory. There's limitations to both of these options. So if we look at the ovarian tissue freezing that I just talked about, because I do these surgeries all the time, the child that has cancer has to go through a laparoscopy for me to recover the ovary. And then when it's time to put the ovary back, they have to go through another whole surgery. And on top of that, we always worry that this tissue that we're taking from young girls with cancer may have malignant cells in it. There have been flow cytometry studies identifying that. And we worry that when we put the tissue back, that we could risk reintroducing malignancy into those patients. So this is not a perfect science. And then with IVF, just talk to any IVF patient about the burden of going through all of those shots week after week. I don't know if you've ever seen on, I don't know if you're Instagrammers, but those babies surrounded by a heart made out of hundreds of syringes for injections. And so when people go through IVF, anyone can tell you about the physical burden and the psychological burden, but that's not all. Actually, patients going through IVF, and this is a huge access to care issue, face a big financial burden because IVF, if it's not covered by your insurance, can be up to $15,000 a cycle. So on top of those limitations, our biggest impediment with our current techniques is the incredibly inefficient process that the ovary naturally is built to run in terms of the maturation of the oocyte. So you can think about it like a truck factory where you have all this machinery in place to make 100 trucks, but the number of trucks that comes off the line that's actually usable might just be 1. And so it's an incredibly inefficient process. And so we need a better way to get these immature eggs to a mature state to make them more usable for our patients. So hopefully that kind of paints the picture of what we're up against. And I'm going to turn this over to Dr. Rios to tell you a little bit about what we're planning.
So as you can see here, both of these techniques have immature oocytes that are often just discarded. And so to talk about what we're planning for this Magee Prize, we have to go back to some ovarian biology, which luckily has already been talked about today. So I'll go a little bit quicker on this. But we are born with all of our eggs. Those are those primordial follicles that are quiescent in the ovary. And the process to get those primordial follicles to an ovulatory follicles takes 6 months, with the last 2 months really being dependent on hormones in our body. And so the last really 2 weeks is when we start introducing those IVF hormones to move what are called antral follicles into those ovulatory follicles. And then we do our egg retrieval. Many people maybe believed, or you don't know, that they thought these cycles of maturation happen in isolation. But no, there's constant asynchronous maturation of oocytes. And so when we go and collect these eggs at the time of egg retrieval, if you focus on that, those pink dots in the middle, we will get immature eggs as well as mature. The immature egg, or the germinal vesicle, is an antral follicle. It's the egg that's in an antral follicle. It can't be fertilized with sperm. The next one is, we've started to mature, but we're not quite there. And that's called metaphase 1. And then the metaphase 2 oocyte is the one that can be fertilized. And so those are the ones we freeze, those are the ones we fertilize with sperm, and the rest get discarded. And so what can we do with those discarded eggs? Or can we even eliminate those 2 weeks of hormones and really just put those germinal vesicles into a dish in the lab and mature them, a process called in vitro maturation, or IVM. We have been studying this in humans and other animal models for decades. The animals have figured this out. They have been using this for reproduction in animal models for a long time. Why is that not the case for humans? And so looking at this animation, a lot of in vitro maturation protocols have really focused on nuclear maturation. And so we can see that nucleus dissolving, chromosomes are separating, this is moving into a polar body. But what's happening in that other bigger circle? And a lot of times we're not focusing on the quality of the egg that is maturing in an in vitro maturation. And so there are a couple different protocols for this. There's the conventional IVM, which is what we've done for decades, focusing on that nuclear maturation, where essentially we take the germinal vesicles, we stick them in a dish, we mature them with hormones and other factors that are similar to what's in the body, and then we can get a metaphase 2. So we see that nuclear maturation. However, we're not getting great maturation rates, maybe about 50%, sometimes 60, and also lower embryo yield and live birth. A newer protocol for IVM kind of pauses that nuclear maturation and allows the cells in the cytoplasm, our organelles, our proteins, to kind of catch up and mature first, and then we move it into that nuclear maturation phase. This is advantageous, potentially in that we're seeing some higher maturation rates in early studies and some improved embryo yield, but it does increase lab burden, and it's very recently introduced, and there's not a lot of science behind it. When we look at IVM research, there's a lot of gaps in this knowledge, and that's why this hasn't moved forward in humans. Our project is going to be to look at oocyte quality from 2 different protocols. We're going to optimize those protocols while we study them, and then we're going to apply those to some of those difficult populations we've talked about. And so looking at the pictures over here on the left side of the screen, ideally when we then can harvest the oocyte for ovarian tissue cryopreservation prior to cancer or other treatments, we can then freeze not just the ovarian tissue, but also some immature eggs, because we can put them in culture and mature them. We could also do this at the time of endometriosis or ovarian cancer surgery in reproductive age women, and potentially also decrease the number of days of injections and maybe offer a more cost-effective multiple cycles, which will decrease disparities in infertility treatment. Our team is combined of 4 team members, so myself and Dr. Flick, we are both experts in IVF and fertility, as well as fertility preservation. And here at UPMC, Dr. Flick mentioned, but we have a unique center called the Center for Reproductive and Transplantation, or the CRT. We process hundreds of ovaries for fertility preservation for pediatric, adolescent, and young adults. And so we will have a great access to tissue for these patients. Dr. Flick, with her experience with endometriosis, will also have a large access to ovarian endometriosis and ovarian tissue that comes along with those surgeries. We also here at the MWRI are working with Miguel Reneo Enriquez, who is a vital member of our team, who is going to be doing basic science testing to look at those oocyte quality. And then we have an international collaborator, Dr. Anderson, who is a pioneer in ovarian tissue cryopreservation, as well as in vitro maturation in ovarian tissue. So our planned experiments, aim 1, is really to focus on the clinical, and we are going to have to look at those nuclear maturation rates between this traditional IVM protocol as well as the biphasic protocols in our discarded immature eggs from IVF. We have a robust amount of these that happen annually in both of our IVF labs. Then we'll be freezing those eggs, the ones that mature, but also the ones that don't mature, and have those looked at under the microscope to look at differences in quality for protein expression, metabolism of the egg, and organelle function. And then our third aim is then to apply the best protocol from aim 1 and 2 to those difficult tissues for ovarian tissue cryopreservation, endometriomas, and oocytes from older ovaries. So one of the things to note is that our reproductive and infertility care is highly underfunded. And so we only get about 3% of the budget from the NICHD. And so it's very hard to study infertility. And so that's why we're really focusing on the oocyte quality here, because we can't focus a lot of times on embryos and study those. And so we're starting with the basic steps of the egg. Our experiments are already in process, and so we've already been able to isolate and mature germinal vesicles here in the UPMC lab at a pretty high rate with the standard traditional IVM protocols. And then we've also been able to freeze these, and we're starting to start our protein expression studies in Dr. Reneo Enriquez's lab. And then we've also been able to isolate immature oocytes from ovarian tissue in the tissue that we're processing. And Dr. Anderson is working on the IVM rates over in Denmark. And he has a lesser maturation rate, but we may expect that from the tissue because they're going to be a little bit different than the eggs that we have from the ovary and IVF. And so looking at oocyte expression models, this here, you can compare these two different oocytes, and we can see that there's different protein expression in the nucleus and the cytoplasm. And so these are some of the studies that we're planning to look at our quality of our eggs. And then we can't have this project without the expertise of Dr. Anderson, and so he does have a few words for our audience.
My name is Claus Yling Andersen from Copenhagen in Denmark. I'm happy to participate in this project with Julie, Rebecca, and Miguel for the Magee Prize. I do think that this project is very interesting and will pave the way for a lot of women undergoing fertility preservation will have augmented chances of having their own biological children. In addition, I do think that this project has a lot of wider implications in the sense that if we can establish a human platform for IVM, then we may avoid ovarian stimulation and the massive cost of exogenous gonadotropins, and therefore generate a assisted reproduction technique which can be used by many more women because it's cheaper worldwide, and there will be a lot more benefiting from it in this sense. So I do hope the committee and the audience will look favorable at our project. Thank you.
We want to acknowledge our teams both here at UPMC and MWRY, as well as at University Hospital in Cleveland, because without our team, these experiments will not be possible. And we want to thank you. We are honored to be up here presenting our research and understanding that infertility is a global health problem, and we need to unlock better, less expensive treatments to provide equity in fertility care. Thank you.